welcome to the deep dive. Today we're venturing into something pretty cool in our genetic code.
Yeah, we're looking at recent findings in the genomes of humans and, our great ape cousins.
Specifically, we're talking about DNA structures that go, well, beyond that classic double helix image.
Exactly, and what they might mean for our health, maybe even our evolution.
And for you listening, this deep dive is your shortcut to some really cutting-edge stuff in genetics. We'll break down these non-traditional DNA structures.
We'll explore why we're really only getting a good look at them now.
Right, and what the first clues suggest about their role in biology. Great ape biology, which, includes us.
Mm-hmm. We're basing this on a fascinating new study just out in Nucleic Acids Research.
Okay.
And it uses these amazing new complete genome maps. They call them telomere-to-telomere assemblies.
Right, the T2T ones.
Yeah, covering humans, chimps, bonobos, gorillas, orangutans.
And the siamang, too, as a comparison.
Exactly, a lesser ape perspective. So our mission today is to really unpack what this non-canonical DNA is, why it's been sort of hidden, and what these initial findings are hinting at.
Okay, let's start there. The double helix, that's what everyone knows. But this study mentions non-B DNA. What are we actually talking about?
Yeah, think of the standard DNA helix as the B form. Non-B DNA is just different shapes. The DNA sequence itself allows it to fold up in unusual ways.
Like DNA origami.
Yeah, kind of. The study mentions a few key types. You've got bent DNA, pretty much what it sounds like, a curve.
Okay.
Then hairpins, where a single strand loops back on itself. G-quadruplexes, or G4s they form in guanine-rich areas. 4 strands involved.
Wow, 4 strands.
And Z-DNA. That one's cool. It twists the other way, left-handed instead of right.
So DNA isn't just this rigid ladder. It can get quite complex structurally, and this happens because of specific sequences, often repetitive ones.
That's the key. Certain patterns, especially repeats, make these alternative shapes more likely to form. And here's the kicker. These very regions, the repetitive ones where non-B DNA likes to form, have been, well, incredibly difficult to study accurately until very recently.
Why was that? What was the problem?
It really came down to the sequencing tech we use for years, mostly short-read sequencing.
Okay.
Imagine trying to assemble a 1,000-piece jigsaw puzzle, but a huge chunk of it is just blue sky. All the pieces look almost the same.
Right. Impossible to know where each identical piece goes.
Exactly. Short reads chop the genome into tiny bits. If lots of bits are identical repeats, figuring out their order and how many there are as well, it was a nightmare. These non-B DNA hotspots were often in those blurry, repetitive zones.
Got it. So the puzzle had missing or smudged pieces, right, where this interesting stuff might be happening. What changed? What let us finally see the full picture?
The big breakthrough is telomere-to-telomere T2T assembly. It relies on long read sequencing.
Longer reads.
Yeah.
So bigger puzzle pieces.
Much bigger pieces. These reads are long enough to stretch across entire repetitive regions, sometimes huge ones. It lets researchers finally build a complete, truly gap-free map of each chromosome, from end to end, telomere to telomere.
And that complete map is what allowed this new study to really explore these non-B structures properly across all the great apes they looked at.
Humans, chimps, bonobos, gorillas, the 2 orangutan species, plus the siamang. Yep.
It's amazing how recent this completeness is. Wasn't the human T2T finished just a couple of years back?
Yeah, the consortium finished the human one in 2022, maybe fully wrapped in 2023. And Katerina Makova, who led this ape study, pointed out that, the original human genome draft left about 8% undetermined.
8%, that's quite a bit.
It is. And it was mostly this tricky repetitive stuff. Now with T2T, those gaps are filled for humans, and this new work does it for the great apes. It's a huge step.
So, okay, complete genomes in hand. The researchers could then predict where these non-B shapes might form. What did they find? What were the sort of headline results?
Well, the big one was that these non-B DNA structures aren't just randomly scattered. They found they are significantly enriched, more common in those very regions that were previously hard to sequence.
The ones filled in by T2T?
Exactly, especially in telomeres.
The protective caps on chromosome ends.
Right, and centromeres.
A central part, crucial for cell division.
Yep. So these unusual DNA shapes are concentrated in really critical functional neighborhoods of the genome.
That seems significant. If they're clustering there, what might they be doing there? What roles does this study suggest?
The paper proposes quite a few potential roles. Things like helping regulate how the genome evolves over long timescales, maybe playing a part in starting DNA replication, the copying process, controlling when genes get switched on or off.
Gene expression.
Right. And maybe even directly involved in the function of the telomeres and centromeres themselves, how those regions actually work.
It's really mind-bending that the physical shape of the DNA could be influencing all these core processes, not just the sequence.
It adds a whole new dimension, doesn't it? And they also touch on health implications.
Yes. You mentioned disease earlier.
Yeah, the introduction notes connections between non-B DNA and certain genetic diseases, even cancer. It's an established link in some cases, though this study was more about the mapping.
So important context.
Definitely. And another interesting observation they made, they looked at gorillas.
Okay.
The gorilla genome actually has a higher percentage of repetitive DNA compared to the other great apes studied.
More of that tricky stuff.
Yes. And interestingly, it also showed a higher number of these predicted non-B DNA sequence motifs.
Huh. So more repetitive DNA, potentially more non-B structures. Is that the idea?
That seems to be the correlation they observed, and it leads to a hypothesis from the researchers that maybe because these non-B structures can be, let's say, a bit less stable, maybe more prone to mutations or breaks, they could be hotspots for things like DNA breakpoints or even chromosomal rearrangements, where bits of chromosomes get swapped around or broken.
Which could drive evolution, but also potentially cause problems, like genetic disorders.
Precisely. It's that double-edged sword idea. Useful for evolutionary change, potentially risky for individual health.
Did they have a specific example of that risk?
They did point to one intriguing case involving satellite DNA near a centromere and zDNA motifs.
The left-handed one.
Yeah. They looked at a region involved in a specific type of chromosome 21 translocation that's linked to a form of Down syndrome.
Okay.
And they found the frequency of potential zDNA forming motifs was, get this, 97 times higher in that specific breakpoint region compared to the rest of the genome.
97 times. That's quite a difference.
It's striking. It strongly suggests zDNA might play a role in the instability that leads to that kind of rearrangement, but, and this is crucial.
Needs more research.
Absolutely. It's a correlation found in the sequence data. Proving that zDNA causes the break requires more experimental work. It's a really tantalizing clue, though.
Definitely sounds like it. So this study is more like creating the map, showing where these structures could be based on sequence.
Exactly. It's predicting the potential. The next big step is experimental validation, actually confirming these structures form in living cells in those predicted spots.
And that might not be straightforward.
Probably not always. Dr. Makova mentions it's likely context-dependent, meaning whether a specific sequence actually folds into, say, a G4 or zDNA might depend on the cell type.
Okay.
Or the organism's developmental stage, what other proteins are around, even chemical tags on the DNA itself, like methylation. It's complex.
So the map is stage 1. Understanding the actual territory in real time is stage 2.
A good way to put it. But this map is invaluable. It really shifts the focus, doesn't it? From just the linear sequence to thinking about the genome's 3D structure as being fundamentally important for function.
It really does.
Yeah.
We needed the technology, the T2T assemblies, to even start asking these questions properly across the whole genome.
Yeah, it's a testament to how technology drives discovery. We're moving past just reading the letters to understanding the architecture. This study really throws open the doors for a lot more research.
So wrapping up this deep dive then, completing these great ape genomes, finally getting those gap-free sequences,
has immediately revealed this hidden layer of DNA structure, these non-B forms.
and they seem to be concentrated in really vital areas, telomeres, centromeres.
Suggesting they could be pretty important players, both in how things normally work and maybe how they go wrong in disease.
Right. Which leads us to a final thought for you, our listener.
Consider this. We focus so much on the A, T, C, and G sequence. But what if the way our DNA folds in 3 dimensions, this intricate origami, we're just starting to map properly?
What if that shape is just as critical as the sequence itself in defining who we are, how our bodies function?
And thinking ahead, what might future discoveries about this DNA architecture reveal about our evolutionary journey? Or perhaps open up completely new ways to think about health and treat human diseases? Lots to think about.